One of the tools we use to understand population genetics is called the Hardy-Weinberg equation. It's an equation that describes the allele and genotype frequencies in a population that is not evolving. Okay, so not in big capital letters. We also call this a population that's in equilibrium. So sometimes we also call this Hardy-Weinberg equilibrium. So in order for a population to be considered in Hardy-Weinberg equilibrium, that is not evolving, there has to be several conditions that the population meets. So first of all, there can't be any new mutations occurring. Natural selection can't be occurring, so no natural selection. The population has to be very large, okay? That population has to be large in order to reduce any chances of sampling errors, right? Random changes happening because there was a small sample size. There can't be any migration, so there is no movement between different populations. And there has to be random mating, so there can't be any bias in the animals or in the organisms and how they choose to mate. And so if all of these conditions are being met, then we would expect that the allele and genotype frequencies should remain the same generation to generation. So the allele frequency, if it was 0.7 now, then it would be 0.7 100 years from now and 0.7 100 years in the past if a population is in Hardy-Weinberg equilibrium. So this is what the Hardy-Weinberg equation looks like. And we're actually going to tend to use these individual components the most. So P is the allele frequency of one allele. Oops. And Q is the allele frequency of the second allele. And then these different components represent genotype frequencies. So P squared, so that 2 is actually squaring. So I guess let me remind you that P squared is equal to P times P. And it's actually, when I calculate that, the genotype frequency of the homozygous for P. Okay, and so for example, if I had P represent a dominant A allele, then it would be the genotype frequency of the big A individual. 2PQ, okay, which is 2 times P times Q, because those are allele frequencies, so remember we could fill in numbers there. And that's equal to the genotype frequency of the heterozygous individuals. So that would be like PQ, or if I'm using my A conventions, big A, little a. And then hopefully you're seeing a trend here. Q squared is equal to Q times Q, and that's equal to the genotype frequency of the individual's homozygous for Q, and in this case, little a, since I'm using the A's, okay? So you can actually use the Hardy-Weinberg equation to solve for different values. So say, for example, you know the allele frequency of just one allele, then you can predict the other allele frequency as well as the 3 genotype frequencies. So we're going to take a look at some examples of that. Okay, so using the HW equation, determine the frequency of the homozygous dominant, heterozygous, and homozygous recessive genotypes if the frequency of the recessive allele is 0.6. Okay, so I've only got one value here. I know that the recessive allele is 0.6. So I'm going to go ahead and set the recessive allele as Q. So Q is equal to 0.6. Remember that we said that allele frequencies have to add up to 1. So P and Q have to equal 1.0. So if I know Q, I can subtract it from 1, and that will tell me P. So 1 minus 0.6 is equal to 0.4, and that's going to equal, P, because we already made Q be 6, okay? And, all right, so the frequency of, so now that I have my P and my Q, I can use my Hardy-Weinberg equation. So remember, I have P squared, 2PQ, and Q squared. So remember that I've set Q as my recessive allele. So that's going to be my homozygous recessive genotype. This is my heterozygous genotype, and this is my homozygous dominant genotype. So now that I've got P, which remember was 0.4, I can take 0.4 and square it, and that is equal to 0.16. And I've got Q. Q is equal to 0.6. So I can take 0.6 and square it, and it's equal to 0.36. And then last one here is the heterozygote, 2 times 0.4 times 0.6, and that is equal to 0.48. And if you add those three numbers up, they add up to 1.0 because, again, you have to reach 100% for your population, right? All of your population is 1.0, okay? So remember that if I'm using the Hardy-Weinberg equation, all of my Hardy-Weinberg assumptions must be true. So no new mutations, random mating, no natural selection, large population size, and which one am I missing? No migration. Okay, so all of those things have to be true in order to use the Hardy-Weinberg equation this way. But at any given moment, we might be able to make those assumptions. So let's do one last one here. It's a little more challenging. So if the frequency of the homozygous dominant genotype is 0.81, what is the allele frequency of the dominant allele, the recessive allele, and then what is the frequency of the other genotypes? So this one's a little bit different from the last one because the information that we're given is different. So instead of knowing the allele frequency, I know what a genotype frequency is. So it's saying the homozygous dominant genotype. So again, using our like A's, right? That's this big A, frequency is equal to 0.81. So remember, that's also equal to, for example, P squared, right? So in order to solve for P, I can take the square root of P squared, right? So P is equal to the square root of 0.81, which is equal to 0.9, right? So by using a little bit of math, I can calculate that P must be equal to 0.9. Once I know what P is, remember I can subtract from 1 to solve for Q. So Q must be equal to 0.1 because 1 minus 0.9 is 0.1, okay? So now I have the frequency of the dominant allele, which is 0.9, and the frequency of the recessive allele, which is 0.1. So that's the first part of the question. Check it off. Smiley face. Did that part. Okay, what is the frequency of the heterozygous genotype? Okay, well, we know our P's and our Q's now. So in order to solve for the heterozygous genotype, which remember is equal to 2PQ, I'm going to take 2 and multiply by 0.9, multiply by 0.1, and that's going to be equal to 0.18, okay? And then the homozygous recessive genotype is little a, and remember that's equal to Q squared. And we solved for Q, which is 0.1, so we're squaring 0.1, which is equal to 0.01. And so just to check my work, I can see that 0.01, 0.18, and 0.81 all add up to 1.0, right? Because, again, to get to that total population size of 1.0. So again, all of those calculations make the assumption that we don't have natural selection, we have no new mutations, we have random mating, no natural selection, large population size, and no migration. In reality, do populations always meet those conditions? No, right? So in reality, we can make the assumption that all populations are undergoing some kind of selective pressures, okay? But if we take like a tiny snapshot, like at this particular, year, 2000, whatever year it's going to be, right? That specific year period, within that period, those pressures might not be very strong. And so we can make that assumption for now, okay? So in reality, populations are probably not in Hardy-Weinberg equilibrium, especially if you look over thousands of years of time. But for like a year period or short periods of time, it can be used as an approximation, right? So it helps us just kind of estimate what a population should be doing. And this is important because we want to be able to compare what Hardy-Weinberg is predicting, okay, to what actually happens in a population. So one of the main uses for this in especially conservation biology is Hardy-Weinberg gives us a prediction for what should happen. So it might be like now or how it should be in the future, okay? So it gives us a prediction that we can then test, okay? So I then go out and measure and test that prediction, and I want to see, does my predicted value match what I saw in reality, so my actual data, okay? If my prediction and my data match each other, then I can conclude that that population is not experiencing any evolution or any of these other things that might be happening in the population. But if my prediction does not match the data, then that means there is some kind of pressure on that population, whether it's natural selection, whether it's migration, whether it's new mutations, whether it's that the population is really small. All of those different conditions of Hardy-Weinberg become possible for me to analyze. Okay, so again, if my data does not match, then 1, 2, 3, 4, 5, one of those 5 conditions of Hardy-Weinberg are not being met. And if I'm a scientist doing research on this area, that's going to be my starting point to ask more questions, like, is this population experiencing migration? Is that why it doesn't match the Hardy-Weinberg frequencies? And so on, going through the different conditions to understand what's happening in that population. And so that's one of the ways we can use Hardy-Weinberg in the real world.